हेलो बच्चों, आज हम ये डिस्कस करेंगे ट्रांसपोर्ट इन प्लांट्स के बारे में। सो व्हाट इज ट्रांसपोर्ट इन प्लांट्स? हाउ डज वाटर रीच द टॉप ऑफ tall trees? So there is no circulatory system in plants. So basically in plants there is no circulatory system. Like animals, there is no circulatory system, blood vessels like this. But how the water reaches to the top of tall trees? So here when you come to plants, so in plants, even though there is no circulatory system like animals, but they have tube-like tissues. So these tube-like tissues are called xylem and phloem. इस xylem और phloem के द्वारा water और food transport होता है. So xylem mainly helps in transport of water and phloem helps in the transport of food. So xylem and phloem both come under vascular tissues, which will transport food and water, and they are made up of various components. So here this is vascular bundle. So inside vascular bundle there is xylem and there is a phloem, through which the water conduction and food conduction takes place. So water conduction takes place in this direction, that is towards upwards. Food conduction takes place in this direction. So here xylem and phloem transport the substances in opposite direction, where xylem will transport water and minerals and the phloem will transport food type substances. Okay. So here when you come to plant transport, it transports various substances like gases, minerals, water, hormones, organic solutes. And this transport, it may be short distance or it may be long distance. So short distance means from cell to cell, one cell to other cell. Whereas long distance means it is from roots to stem or root to leaves. So this transport of substances, either it may be short distance or long distance. And these substances are transported in large quantities. So they are transported in large quantities. That is called bulk. So bulk means they are transported in large quantities. So this bulk movement of substances through xylem and phloem, it is called translocation. As bulk, large quantities will move, it is also called mass or bulk flow system. Okay. Here substances are moving from one point to other point in large quantities. So what is the driving force for this? So the driving force for this is the pressure differences between the 2 points. So the 2 points means here इसमें एक पॉइंट आता है कि सोर्स, वेयर इट इज जनरेटेड, सिंक, वेयर इट इज यूटिलाइज्ड। और दो पॉइंट्स में एक पॉइंट सोर्स और दूसरा पॉइंट को क्या बोलता है कि सिंक। सोर्स द जनरेशन ऑफ द सब्सटेंस, सिंक द यूटिलाइजेशन ऑफ द सब्सटेंस। Between these two points there is a difference in the pressure, or it may be difference in the concentration. Then only the substances can move from source to sink. Otherwise it's not possible. So in translocation, either the substances has to move from one point to other point, that is from source to sink, means there should be a pressure difference. So this mass flow of substances from one point to other, it is called translocation. And if you look into the direction of this translocation, so when you take water, so water is from, that is the roots to the leaves. So this water which moves from roots to leaves, it is unidirectional. But whereas when you come to organic solutes means the food substances. So the food substances, the movement of food substances is multidirectional. So here you can see this is leaf, where it is source for food materials, where food is manufactured. So it has to move in this direction as well as it has to move in this direction also. So it is multidirectional. The movement of food is multidirectional, whereas the movement of water is unidirectional. So this is how the water, the minerals and organic solutes, the directional movement. Okay. So here, whatever that substances are transported, maybe suppose if you take water, so water will, along with mineral ions, that is inorganic elements, so water along with mineral ions. So this water and mineral ions, they are taken from soil. So plants ये मिनरल्स और वाटर को सॉइल से लेता है, and these minerals and water which are taken from soil, they has to enter into plant cell, that is they has to enter into the cell. So they has to enter into cell means obviously they has to cross cell membrane, that is they has to cross the plasma membrane, which is selectively permeable membrane. So plasma membrane is a selectively permeable membrane which allows only the required substance. It doesn't allow all the substances. So plasma membrane is made up of phospholipids and proteins. Okay. So how they will move? How they will cross the cell membrane from one cell to other cell? There are different ways, like basically diffusion, facilitated diffusion, and active transport. So this diffusion and facilitated diffusion both can be kept under passive transport. So diffusion and active facilitated diffusion can be categorized under passive transport, where no energy is required for this transport. इसको लिए एनर्जी नहीं चाहिए, लेकिन एक्टिव ट्रांसपोर्ट को एनर्जी चाहिए. सो एनर्जी इज रिक्वायर्ड. So wherever energy is required, it is called active transport. Wherever energy is not required, it is called passive transport. A diffusion और facilitated diffusion में energy नहीं चाहिए, इसलिए वो passive transport आता है. और active transport where energy is required. So here first, when you take in transport in plants, it can be divided into short distance transport and long distance transport. Short distance means from one cell to other cell. That is from cell to cell. इसमें दो टाइप्स रहता, पैसिव और एक्टिव ट्रांसपोर्ट. So long distance transport, that is from one point to other point, that is from roots to leaves or leaves to roots. इसमें mass flow रहता, इसमें mass flow और bulk flow रहता through the xylem and phloem. So water transport through xylem and food transport through phloem. So पहले हम डिस्कस करेंगे पैसिव ट्रांसपोर्ट के बारे में. So पैसिव ट्रांसपोर्ट में इसमें चार टाइप्स आते हैं. पहला है कि सिंपल डिफ्यूजन, फैसिलिटेटेड डिफ्यूजन, ऑस्मोसिस और इम्बाइबिशन. सिंपल, फैसिलिटेटेड, ऑस्मोसिस और इम्बाइबिशन. पहले हम डिस्कस करेंगे डिफ्यूजन के बारे में. पहले इसमें डिफ्यूजन में आए तो डिफ्यूजन इज द मूवमेंट इज इन रैंडम. सो रैंडम बोले तो कोई स्पेसिफिक डायरेक्शन नहीं रहता. सो डायरेक्शनलेस, नो स्पेसिफिक डायरेक्शन. So in diffusion, the molecules will move from high concentration to low concentration. So the molecules will move from high concentration to. So here the driving force for this is the concentration difference. इसमें एक पॉइंट से दूसरे पॉइंट को concentration difference रहना. और ये प्रोसेस में एनर्जी नहीं चाहिए, नो यूज ऑफ एनर्जी. सो इसको ये मूवमेंट को पैसिव बोल सकते हैं. और इट इज वेरी स्लो प्रोसेस. और ये डिफ्यूजन प्रोसेस लिविंग सिस्टम्स और नॉन लिविंग सिस्टम्स बोथ में हो सकता है, दोनों में हो सकता है. And mainly it takes place in gaseous movement within the body. A plant body में gaseous movements के लिए diffusion चाहिए. क्या gaseous movement रहता है plants में? CO2, oxygen, carbon dioxide और oxygen movement का रहता है. Oxygen coming out from plants during photosynthesis. CO2 released during respiration, O2 taken during respiration. A diffusion में a diffusion rate रहता, diffusion rate बोले तो the rate of movement of substances. So it is affected by various factors. So the first factor is concentration gradient. So gradient बोले तो difference from one point to other point. Temperature, pressure, permeability of the cell membrane. So cell membrane plays an important role. Our size and solubility of the substances. सब्सटेंसेस का साइज और उसका सॉल्युबिलिटी में डिपेंड होता है. अब देख सकते हैं डायग्राम में. In this water, a blue color dye is added, and gradually this dye is moving to all the parts of the water. So this is a diffusion, where it spreads slowly, no requirement of energy, a passive process. So अभी तक हमें डिस्कशन किया डिफ्यूजन के बारे में. So बात में आएंगे फैसिलिटेटेड डिफ्यूजन. फैसिलिटेटेड डिफ्यूजन, इसमें ए बी सेल मेम्ब्रेन को क्रॉस करना है. सब्सटेंसेस दे हैज टू क्रॉस सेल मेम्ब्रेन. That is from high concentration to low concentration. But here there are separate specialized transport proteins are involved. So इसमें दो रहता, that is carrier proteins और channel proteins. Specialized transport proteins में carrier proteins रहता और channel proteins रहता. और ए प्रोसेस में एनर्जी नहीं चाहिए, नो नीड ऑफ एनर्जी, इसलिए इसको क्या बोलता है कि पैसिव ट्रांसपोर्ट. एंड द ड्राइविंग फोर्स फॉर दिस इज कॉन्सेंट्रेशन ग्रेडियंट, द डिफरेंस इन द कॉन्सेंट्रेशन. सो फैसिलिटेटेड डिफ्यूजन में the movement of molecules across the cell membrane through specialized transport proteins. A specialized transport proteins ये plasma membrane में रहता है. So these specialized transport proteins are present in plasma membrane. ये प्लाज्मा मेम्ब्रेन में रहने वाले प्रोटीन्स ये फंक्शन करता है, द ट्रांसपोर्ट. और फैसिलिटेटेड डिफ्यूजन में प्रोटीन्स का इन्वॉल्वमेंट होता है, इसलिए ये प्रोटीन्स के इन्वॉल्वमेंट कब इसलिए ये रहता, और ये इसमें आता है कि सैचुरेशन. Saturation means when all the proteins are involved in the transport, so there is no further transport. Or a protein specific रहता, they are very specific, which will take selected substances. Or a proteins they are sensitive to inhibitors. कुछ substances रहता which inhibit the activity of the proteins. और A proteins में some channels are always open and others are controlled. Controlled means there will be opening and closing depending upon the need. Controlled means इसमें opening or closing रहता, which is dependent on need and necessity. इसलिए ए फैसिलिटेटेड डिफ्यूजन में प्रोटीन्स आर इन्वॉल्व, दे आर वेरी स्पेसिफिक, दे आर सैचुरेटेड, सेंसिटिव टू इनहिबिटर्स, एंड दीज चैनल प्रोटीन्स some are always open, some are controlled. Controlled means which are opening and closing. और इसमें फैसिलिटेटेड डिफ्यूजन में पोरिन्स और एक्वापोरिन्स रहता। सो पोरिन्स कहाँ रहता है? Porins are proteins which are huge pores present on outer membrane of plastids, mitochondria and bacteria. So plastids बोले तो हमें ले सकते हैं क्लोरोप्लास्ट। So chloroplast. Okay. To allow some specific size of proteins. Or aquaporins. Okay. So these are protein channels which are meant for transfer of water, and in few cases it is small solutes. So ट्रांसपोर्ट के वाटर के बारे में है तो ये बाई डायरेक्शनल रहता। बाई डायरेक्शनल रहता है तो वाटर अंदर मूव फ्रॉम inside to the cell, move from outside the cell. So porins or aquaporins दोनों facilitated diffusion में होते हैं। Aquaporins are meant for transport of water. Porins meant for the transport of small proteins. Through these porins, the small proteins are transported. So आप देख सकते हैं डायग्राम में। So it is outer side of the cell. This is plasma membrane. This is the transport protein through which water is moving. Okay. And here, so the molecules are transported from outside to inside. So diffusion, what is the importance of diffusion? Why it is required? So facilitated diffusion is specifically meant for movement of polar molecules and large ions. Okay. So which are dissolved in water but cannot cross through the plasma membrane. Why they cannot cross the plasma membrane? The reason is the phospholipid tile, the tile of, that is when you take the phospholipid tile which is hydrophobic in nature. Hydrophobic बोले तो which reflect water, which hate water. Okay. So a hydrophobic tile के वास्ते only non-polar molecules like oxygen, carbon dioxide can diffuse the membrane. So for the movement of polar molecules and large ions, so they require the proteins, which is done in facilitated diffusion. So large molecules are transported by transmembrane, that is the proteins. Thus proteins are called permeases, such as permeases. Okay. और a facilitated diffusion में okay, इसमें 3 टाइप्स रहता है: यूनिपोर्ट, एंटीपोर्ट, सिम्पोर्ट. Okay. Uniport means only one molecule moves. Only one molecule. और एंटीपोर्ट और सिम्पोर्ट में 2 molecules will move. और एंटीपोर्ट में thus 2 molecules will move in opposite direction. और सिम्पोर्ट में आए तो the 2 molecules will move in the same direction. Either this or either this. Symport means the same direction. Antiport means opposite direction, 2 molecules. Uniport means only one molecule will move. So these are the 3 types of facilitated diffusion: uniport, antiport, symport. सब देख सकते हैं पिक्चर में, इन ई पोर्ट ओनली वन मॉलिक्यूल इज ट्रांसपोर्टेड. Symport, two molecules are transported in the same direction. Antiport, two molecules, one in both in opposite direction. Okay. So अभी तक हमें डिस्कशन किया डिफ्यूजन और फैसिलिटेटेड डिफ्यूजन. अभी हमें आएंगे एक्टिव ट्रांसपोर्ट के बारे में. Active means itself says it uses energy. Okay. So for this active transport also the membrane proteins are required, okay, which play a major role, okay, in facilitated as well as active transport. And in this active transport, the substances will move from low concentration to high concentration. So they will move from low to high concentration. Here also, like facilitated diffusion, it reaches to saturation. And they are also very specific. They will carry the specific type of substances. And they are sensitive to inhibitors. So the proteins which carry the substances, they are sensitive to inhibitors. So here in active transport, the substances will move from low to high concentration, which is called uphill. Whereas in diffusion and facilitated diffusion, it is downhill. Downhill means from high to low concentration. So here you can see energy ATP is used. So here it is low concentrated and here it is high concentrated. Even then the substances will move from low to high concentration. Here you can see this is passive transport with diffusion, facilitated diffusion. So where substances move from high to low, or active transport from low to high, which is called uphill. Whereas it is called downhill. From high to low concentration, it is called downhill. From low to high, it is uphill. Okay. So when you look into the differences between transport to the diffusion and facilitated diffusion. So diffusion, just concentration gradient. There is no other one. They don't require any carrier molecules. It is not sensitive. Energy is not required. No saturation. But when you come to facilitated diffusion, okay, इसमें common point है कि concentration gradient दोनों में. but इसमें चाहिए special proteins sensitive to inhibitors, proteins. Energy is not required. It is also common point between diffusion and facilitated diffusion. But here saturation रहता when all the proteins are involved in this process. Okay. So अभी तक हमें डिस्कशन किया पैसिव ट्रांसपोर्ट और एक्टिव ट्रांसपोर्ट की. So here there is a relation between plant and water. So why it is related? The water is essential for all physiological activities of the plant. So without water, physiological activities cannot be performed. Whatever it is, either respiration or photosynthesis, they require water. So water is the medium for most of the substances which are dissolved. और protoplasm और cytoplasm में maximum it is made up of water. 90% it is made up of water. Where different molecules are dissolved in the water. Okay. So the availability of soil water to plants depends on quantity of water stored and its relationship to soil water potential. So here we'll get a new term, that is water potential. What is this water potential? So water potential means in water there is an energy, that is potential energy, which helps in movement of water. So the free water molecules which are present in water will have the potential energy which is used for movement of this total free energy present in the free water molecules, it is called water potential, which is meant for to do work. Work means here the movement of water. So it is the measurement that combines the effects of, that is, it is water potential, it is a combined effect of solute concentration and pressure. It is dependent on majorly two factors: the solute concentration and pressure. So pure water which has more free water molecules, that's why it has the greatest water potential. So in प्योर वाटर, सो प्योर वाटर में ज़्यादा फ्री वाटर मॉलिक्यूल्स रहता, इसलिए इसमें मैक्सिमम वाटर पोटेंशियल रहता, and it is represented by symbol psi. The units of the water potential are expressed in pascals. So इसमें the water potential of pure water is 0. So water will always move from high water potential to low water potential. So downhill, here it is, it will move from high to low water potential. So here water potential plays an important role in the movement of water. So water always will move from region of high water potential to region of low water potential. और ये वाटर पोटेंशियल, ये दो फैक्टर्स पे इफेक्ट होते हैं, एक है कि सोल्यूट कॉन्सेंट्रेशन और दूसरा है कि प्रेशर. हमें डिस्कस करेंगे ये फैक्टर्स के बारे में. सोल्यूट, सो ए सोल्यूट पोटेंशियल, so when we add solute to a solution, that is, when we add a solute to solution, so what happens is, due to addition of this solute to the solution, the water potential of the solution will decrease. Okay. Why it will decrease? So the solute molecules will bind with free water molecules. सभी वाटर में क्या होता है कि फ्री वाटर मॉलिक्यूल्स का नंबर कम होता है. Due to decrease in free water molecules, obviously the water potential will decrease. That is, as we go on adding solute to solution, the water potential of the solution will decrease. So this decrease in water potential on adding solute, it is called solute potential, or otherwise it is also called osmotic potential, and it is represented by the symbol psi s. and here as it is decreasing, always the value of solute potential is negative. So इसमें solute potential का वैल्यू नेगेटिव रहता, कभी भी नेगेटिव रहता, क्योंकि solute ऐड करने के बाद ये water potential डिक्रीज होता है. So here this solute potential with addition with pressure potential gives the value of water potential. So water potential is a sum of solute potential plus pressure potential. Psi P means pressure potential, psi S means solute potential, which is sum of these two values will give rise to water potential. Okay. So here water which is present in the protoplasm, 90% of the protoplasm is made up of water. So this water will exert a pressure against the cell wall. So protoplasm cell का अंदर रहता, cell wall ऊपर रहता. So the water which is present in the protoplasm will exert pressure on the cell wall in this direction from inside to outside. So this pressure is called turgor pressure. Okay. Or a cell wall both rigid structure. So this rigid structure which will exert again the pressure on the protoplasm in this direction. So A direction में pressure रहता. So इसको क्या बोलता है कि the opposite pressure to turgor pressure, इसको क्या बोलता है कि wall pressure. A turgor pressure is due to protoplasm, that is water present in the our air wall pressure, that is from cell wall in this direction. So both the values are equal, but the direction is opposite. So when you take the values of turgor pressure and wall pressure, both the values are equal, but their directions are opposite. So a cell which experiences turgor pressure, it is called turgid cell. इसको क्या बोलता है कि turgid cell. Turgid cell बोले तो ये सेल में turgor pressure रहता. turgor pressure का क्यों रहता? Due to sufficient amount of water. आपके डायग्राम में देख सकते हैं ये turgor pressure है in this direction, a wall pressure in this direction. Okay. Both are opposite in direction. Turgor pressure and wall pressure are opposite in direction, and but the values are same. So this is a turgid cell. And here water enters into the cell by a process called osmosis. By a process called osmosis. Okay. So here this cell wall pressure is due to presence of cell wall. A cell wall के वास्ते wall pressure रहता. Okay. So this turgor pressure is developed due to entry of water into the cell. और ये turgor pressure का use भी रहता plants में. It is responsible for extension growth in cells. It is useful for extension growth in cells. So अभी तक हमें डिस्कस किया solute potential और osmotic potential और turgor pressure and wall pressure. हमें अभी डिस्कस करेंगे pressure potential के बारे में. So pressure potential is the pressure exerted by cytoplasm of the cell due to entry of water. Due to entry of water, the pressure potential will increase. That is, the value of pressure potential is always positive, whereas the value of solute potential is always negative. ए प्रेशर पोटेंशियल के वास्ते क्या होता है कि द वाटर पोटेंशियल विल इंक्रीज. प्रेशर पोटेंशियल के वास्ते वाटर पोटेंशियल इंक्रीज होता है, इसलिए इसका वैल्यू पॉजिटिव रहता, कभी भी पॉजिटिव रहता. और सोल्यूट पोटेंशियल का वैल्यू और ऑस्मोटिक पोटेंशियल का वैल्यू कभी भी नेगेटिव रहता. किसके लिए? सोल्यूट के वास्ते वाटर पोटेंशियल डिक्रीज होता. okay. और कुछ केसेस में प्रेशर पोटेंशियल नेगेटिव रहता है, इज नेगेटिव इन प्लाज्मोलाइज्ड सेल. एंड जाइलम वेसल में प्रेशर पोटेंशियल नेगेटिव रहता, टर्जिड सेल में पॉजिटिव रहता, और फ्लैसिड सेल में इसका वैल्यू 0 रहता. सो प्रेशर पोटेंशियल कभी भी पॉजिटिव वैल्यू रहता, लेकिन जाइलम वेसल और प्लाज्मोलाइज्ड सेल में इसका वैल्यू नेगेटिव रहता. टर्जिड सेल पॉजिटिव और फ्लैसिड सेल में इसका वैल्यू 0 रहता. So water potential is affected by solute potential and pressure potential. अभी हमें डिस्कस करेंगे ऑस्मोसिस के बारे में. ऑस्मोसिस बोले तो क्या है? So osmosis is a type of diffusion, only water molecules will move. So इसमें सिर्फ water molecules का movement रहता. That is from high water potential to low water potential through semi-permeable membrane. Semi-permeable membrane बोले तो cell membrane और plasma membrane. Okay. So this movement of water in osmosis depends on the pressure gradient and concentration gradient, the difference in the pressure and the difference in the concentration. Okay. So water has to move by osmosis means the 2 points, that is from one point to other point, the 2 points should have a concentration difference and pressure difference. Okay. So इसमें दोनों पॉइंट्स में सेम रहता तो वाटर पोटेंशियल, इफ इट इज सेम एट 2 points, they are at equilibrium. Equilibrium means the net movement is 0. So the net movement of water is 0. So osmosis takes place through semi-permeable membrane. So and only it allows solvent molecules, no solute particles. और ये osmosis it takes place from high water potential to low water potential through semi-permeable membrane. और ये प्रोसेस को स्टॉप करने के लिए if you apply force from the solution side, opposite force, that is osmosis ये डायरेक्शन में रहता है को अब if you apply force on this in this direction, which will stop this process, that is called osmotic pressure. Osmotic pressure different है और osmotic potential different. Okay. Osmotic pressure is the pressure applied to stop the process of osmosis. आप लेफ्ट साइड में डायग्राम में देख सकते हैं, there are 2 solutions, a dilute solution or a concentrated solution. A solute molecule से इधर, this is semi-permeable membrane. Here the osmosis will take place from this dilute solution to concentrated solution. That is, here the water potential is high. Here the water potential is low. This is Thistle funnel experiment to demonstrate the process of osmosis. So इसमें हमें देख सकते एक बीकर में water लिए. A thistle funnel, it is tied with semi-permeable membrane और इसके अंदर सुक्रोज solution बोले तो concentrated solution. Water, it is a diluted solution. Due to osmosis, water will move into this sucrose solution in this direction. That is, osmosis is taking place in this direction. To stop this process, we should apply the osmotic pressure in opposite direction in this way. This is osmotic pressure, so that the process of osmosis is stopped. See there, osmotic pressure or osmotic potential ka differences. There are two or quite different terms which look like same but they are different. Osmotic pressure is the pressure which is applied to osmotic system to prevent the entry of water. Or osmotic potential है कि which will decrease the water potential, that is, which will decrease the free energy of water. For osmotic pressure, it can be developed only in closed system. और इसमें osmotic potential closed और open both systems में develop होता है. Osmotic pressure का value positive रहता और osmotic potential का value कभी भी negative रहता. ये कि differences between osmotic pressure and osmotic potential. और ऑस्मोसिस में दो टाइप्स रहता. एक है कि एंड ऑस्मोसिस और दूसरा एक्स ऑस्मोसिस. End osmosis means water will flow into the cell. X osmosis means water will come out of the cell. Okay. So when will the water will flow into the cell? In which condition the water will come out of the cell? Okay. If we keep the raisins in water, the dried grapes, after some time the raisins will get swelled due to absorption of water, due to entry of water into this. That is, this is the water enters into the cells, which is end osmosis. Exosmosis, where water comes out of the cell, when it will take place. So when it is kept in a solution, that is where the solute concentration is high. That is when you keep grapes in a concentrated sugar solution. एक grapes में रहने वाले water बाहर आते हैं. Okay. So these are the differences between diffusion and osmosis. Diffusion mostly in gases, very less in liquid, very rare in solids. Whereas osmosis is limited to liquid medium. A diffusion may do not require semi-permeable membrane, whereas osmosis में semi-permeable membrane चाहिए. Diffusion में solute और solvent both move हो सकता है, लेकिन osmosis में सिर्फ solvent molecules diffuse. It depends on concentration gradient. It depends on solute potential. Okay. So here only solvent molecules movement, here all particles movement. Okay. So the types of solution में आए थे. What is this types of solution? Based on their concentration of water, there are three types of solutions. So isotonic, hypertonic, hypertonic. Iso means same, So when the concentration of water is same on both points, when there is same, there is no entry of water, exhaust water from the cell. That is, when the concentration of solution surrounding the cell and the cell both are having same concentration, there is no exit or entry of water. Hypertonic बोले तो hypertonic is a solution which has lower solute concentration. इसके वास्ते क्या होता है कि water will enter into the cell. Hypertonic which has high solute concentration. So इसमें क्या होता है कि water will come out of the cell where you can see X osmosis. Here you can see end osmosis when it is placed in hypertonic solution. So a picture में देख सकते animal cell, plant cell when kept in 3 solutions. When animal cell is kept in hypertonic solution, so water will enter into the cell. After this, some time the animal cell will burst due to lack of cell wall. Like in a plant cell में it will not burst due to presence of cell wall, it will become turgid. So when it is kept in isotonic solution, so it is in equilibrium, the net movement of water is in equilibrium. But when you keep in hypertonic solution, the water is lost from the cells, where the cells will be shriveled. Okay. Whereas when you come to plants, it is called plasmolized, where the cytoplasm shrinks. So here you can see. So when a cell is placed in hypertonic solution, so the water is lost from the cells. So where this protoplasm shrinkage takes place. And this process is called plasmolysis. And if the same process continues for longer period, it leads to death of the cell. So here it becomes flaccid. That is, the turgor pressure is 0. Tp is 0. Obviously, wall pressure will also be 0. But again, when you keep the same cell in hypotonic solution, that is dilute solution, water will enter into this. Again, it comes to normal state. That means it becomes turgid. So this is called deplasmolysis. So इसको क्या बोलते हैं कि deplasmolysis. So this is about the water potential, solute potential, pressure potential, turgor pressure, wall pressure, and the 3 types of solutions. So that's all for today's session. Follow us on SPSVERSITY. Thank you all. We'll meet again in the next session.